Protein synthesis is a process that links amino acids into a polypeptide chain using the information found in DNA. It consists of two parts, translation and transcription, and takes place in the nucleus.
Transcription factors attached to the promoter region. It creates a transcription initiation complex. RNA polymerase II lands on the promoter. RNA polymerase binds to the promoter. DNA strands unwind. Polymerase initiates RNA synthesis start point on template strand.
Begins as RNA polymerase moves along DNA. Continues to untwist double helix. RNA polymerase II reads sense DNA, template strand. Adds nucleotides growing mRNA in 5' to 3' direction.
Occurs when RNA polymerase II reaches DNA stop region or termination sequences in DNA. RNA polymerase continues 10 to 30 bases past sequence and releases pre-mRNA. RNA polymerase jumps off DNA and mRNA peels completely free from sense DNA.
Must occur before pre-mRNA leaves the nucleus. 5' modified guanine cap is added to 5' end of mRNA. 3' poly A tail is added to 3' end. Cap and tail are important for moving transcript out of nucleus and help protect mRNA from degradation by hydrolytic enzymes. Help ribosomes attach to the 5' end of the mRNA.
Process of removing introns from pre-mRNA. mRNA is edited by spliceosomes. Spliceosomes contain snRNAs plus various proteins. snRNAs attach to each end of the intron, while the other sRNAs pull the ends of the introns. It removes introns and it connects exons.
Used to determine which mRNA codon codes for which particular amino acid. 3 codons do not indicate amino acids but are stop signals, marking the termination of translation.
Actual synthesis of polypeptide occurs under direction of mRNA. It takes place on ribosomes.
Ribosomes facilitate specific coupling of tRNA anticodons with mRNA codons during protein synthesis. Consists of 2 subunits, large and small. Each consists of ribosomal RNA, made by RNA polymerase and various proteins, made in nucleolus inside nucleus. Each has binding site for mRNA and 3 binding sites for tRNA molecules. The P site holds tRNA carrying the growing polypeptide chain. The A site carries tRNA with next amino acid to be added to chain. Discharged tRNAs leave ribosome at E exit site.
tRNA molecule consists of a strand of about 80 nucleotides that fold back on itself to form a 3D structure, a rough L shape. Includes loop containing anticodon and an attachment site at the 3' for an amino acid. Made along genes in nucleus by RNA polymerase III.
In cytosol that connects proper tRNA to its correct amino acids. Ribosomes move tRNA with attached polypeptide form A site to B site. mRNA and tRNA are shifted from A to P and from P to E. tRNA in E site is free to float away to pick up another same kind of amino acid.
Consists of series of 3-step cycles as each amino acid is added to preceding one. A site is empty, so tRNA molecule carrying second amino acid lands and fills up the A site. tRNA anticodon must be complementary to mRNA codon in the A site to make sure the correct amino acid is being added. Peptide bond formation occurs as a bond between polypeptide in the P site joins new amino acid in the A site. It separates tRNA at the P site from the growing polypeptide chain and transfers chain to the tRNA at the A site.
Begins when 5' end of mRNA attaches to small ribosomal subunit at start codon. An initiator tRNA carrying methionine amino acid will, with anticodon UAC base pairs with start codon in P site. The large ribosomal subunit attaches. The arrival of large ribosome occurs.
Occurs when ribosome reaches a stop codon on our mRNA. Protein release factor, not tRNA, binds to stop codon. Polypeptide breaks away. Ribosomal subunits separate.
Consists of an amino group, carboxyl group, and a central carbon and R group.
tRNA in large ribosomal subunit acts as ribosome, like an enzyme, to join amino acids to form a peptide bond through dehydration synthesis.
Consists of many ribosomes translating a single mRNA molecule simultaneously. Allows proteins, polypeptides, to be formed quickly.
Primary, sequence of amino acids in polypeptide chain determined by DNA. Secondary, loops, coils, and sheets that frame by hydrogen bonds between non-adjacent carboxyl and amino acid groups. Tertiary, three form held together by interactions between R groups and water caused by electrical charges. Quaternary, three-dimensional structure of a multi-subunit protein and how the subunits fit together.
After translation, polypeptide chains undergo modifications. Sugars, lipids, or phosphates may be added onto certain amino acids. Some amino acids might be cut off. Fascinating!